I'm Ashley Kuhn.
I'm Eben Prostak.
I'm Francesca Rudy.
I'm Spencer Vogel.
And we are Team 8, Polymer Matrix Expansion.
We'll start by giving a brief overview on the scope of our project. Expansion microscopy is a highly attractive method for imaging fixed tissues on the nanoscale. First, it creates a ghost image of a biological sample by removing biomolecules and leaving fluorophores in their place. The fluorophores are used as markers within the cell for its imaging. Then, the sample is expanded about 4 to 5-fold in all directions with the use of a polymer matrix. This allows the sample to be imaged using a cost-effective fluorescent microscope instead of an expensive microscope, which is generally out of budget for most small-scale labs. The current materials used require about an hour or more to expand, so the project's goal is to reduce this time and design a water-swelling polymer that can achieve a similar level of expansion in less than 15 minutes.
In the description of our project, we were introduced to a few design requirements to fulfill. As stated, we were tasked with achieving a 4 to 5-fold expansion of the polymer system in just 15 minutes. We were required to design an experiment to create the polymer system and develop a means of quantitative analysis for the swelling volume in three-dimensional space. Along with the time requirement, it was desired that the expansion of the system be isotropic. This means that the polymer sample should expand at the same rate in all directions. Another design requirement that we were tasked with was finding chemical components, especially monomers, that are low cost and also safe to handle in the lab. Monomers, which link together to form chains in polymers, can be toxic to humans and the environment. Because of this, it was desired that we research less toxic but also cost-efficient monomers for the polymer system.
Our solution was to do lots of research to try and find a hydrogel system that would swell 4 times faster than sodium polyacrylate in water. While expansion microscopy is a very new field, the superabsorbent polymers we were looking to use have many other applications, so we were able to create experiments to test swelling rates in the lab. We found sources that suggested that degree of cross-linking, increasing the pH of the solvent, and using a solvent system with no sodium could increase the swelling rate significantly. While we hoped to test all these variables in the lab, we were only able to do one simple experiment where the degree of cross-linking was changed. Our tests did not show a statistically significant difference in the swelling rate for hydrogel samples with different amounts of crosslinkers, and the samples expanded anisotropically. However, although we were disappointed with the results that we got, our team hopes that more work will be done with the exciting technology of expansion microscopy at UConn in the future, starting with the foundation that our team has created.
To give a broader context on our project, reducing the time it takes for biological samples to expand makes expansion microscopy a more attractive alternative for smaller labs. The direct results of this means that more startups and smaller labs have access to nanoscale imaging that the larger labs do, but for a fraction of the cost.
One important aspect of this project that cannot be forgotten is the potential impact of the application of superabsorbent polymers in expansion microscopy. Possible applications of all fields of agriculture, medicine, science, wastewater treatment, and many others. Just to give a few examples, in the agricultural field, superabsorbent polymers can be placed inside of the soil so that they can absorb water from the rainfall, and then after it stops raining, those polymers have the ability to retain and release that water over a certain amount of time. So doing this can actually help the efficiency of the water and the man hours and growing of the vegetables. This process happens through an osmotic pressure difference, and it can even help improve the growth rate of different types of plants and vegetation. There's also several applications of superabsorbent polymers in expansion microscopy in the medical field. A lot are in drug delivery. The polymer is synthesized with a sugar-responsive gel that can detect drops or spikes in insulin levels in diabetics so that they can monitor the insulin in the pancreas and see whether it needs more or it needs less or something along the lines of that. So there's a ton of applications. I'm sure everybody's really excited to see where expansion microscopy goes. I want to thank everybody for taking the time to watch this video. I hope everybody enjoyed.